Hello everyone, myself Mansi Patel. I am working as assistant professor in Department of Pharmaceutics and Pharmaceutical Technology in Koala College of Pharmacy, uh, which is a constitute college of a Chalura Vidyamandal University. So here I am going to explain you a small topic which is present in pharmaceutical biotechnology subject, which comes in a semester 5. The small topic was in unit 1, genetic engineering. So what is genetic engineering? Genetic engineering is a modification of a gene which is a direct manipulation of any organisms or a living organisms or genome, which is by using biotechnology. Means any kind of alteration we do or a manipulation we do in a gene by using of biotechnology is called as a genetic engineering. So it is the set of a technology used to change the genetic makeup of a cell, including transfer of a genes within and across the species boundaries to produce improved or a novel genes. So novel genes, then after the tools which we are using in genetic engineering. So of course the desired gene which we want to manipulate or alter, then after we need of a restriction endonuclease which act as a biological seizure or molecular seizure, which cut the double stranded DNA and from the limited sites, and also at the same time cutting off a plasmid which is present from a bacteriophage or a yeast cell. Then after we need a DNA ligase which join the cutted strand of a DNA with the plasmid. Then we need a DNA polymerase. The DNA polymerase will produce a new synthesized strand of a DNA complementary to the existing DNA or RNA new template. Then after cloning vectors means the mutation will occur. The once we produce the new DNA strain, the recombinant DNA strain or RNA strain with the help of a plasmid. Then after it will clone present in a cloning vectors is present in a host cell, bacteria or a bacteriophage, where the mutation will be occur. So here I pasted the picture for better understanding. Donor cell, we have a donor cell, and at the same time we have a bacterium cell or a bacteriophage, where from the donor cell we get the gene of interest which we want to alter or manipulate. Then on the other hand we have a plasmid we can take plasmid are going to take, and then after with the help of molecular enzyme, means the molecular seizure or a biological seizure which cut the enzyme from the and ages, and with plasmid also. Once the plasmid and the DNA will the gene which we want to manipulate will be cutting, then after it will be uh, joining with the help of DNA ligase, and at the end we got a new recombinant plasmid, and then a new recombinant plasmid then after added into the bacteriophage or a yeast cell. Then from the bacteriophage and a yeast cell, new mutation genes, means new mutated genes of this recombinant DNA plasmid we get. And then after for the filtration process which we fermentation process we go through, and in the fermentation process the gene will be fermented. So here the principle of genetic engineering: a small piece of a DNA called an plasmid, which is extracted from the bacteria or a yeast cell. As I said earlier, this would be bacteria or a yeast cell, and the small piece of a circular DNA is called as an plasmid. A small section then cut out of a circular plasmid by restriction enzyme as a biological seizure or a molecular seizure. Then the gene from a human insulin is inserted into the gap in a plasmid. This plasmid is now genetically modified. As I said earlier, this plasmid is now genetically modified, and then will be introduced it into the bacteriophage and a yeast phase. I explained these things earlier also. This cell then divided rapidly and start making insulin. Once the insulin is making start, it will be added into the large fermentation vessels to increase the growth of bacteria growth and increase the as well as the production of insulin also. Once the fermentation process is complete, the mixture is filtered and release the insulin, and then after released insulin we are going to purify and pack into the bottles and insulin pens for the diabetic patient, insulin dependent diabetic patients. So in this picture I have explained it more, explaining it in more detail, that we have a plasmid bacteria. Then from the bacteria we get a plasmid, and there's the human cell with containing DNA gene of interest. Insulin gene will be taken, cutting it from the edges, and then after the combined insulin gene with the plasmid DNA, with the help of DNA ligase, the gene of interest will be formed as a recombinant DNA plasmid. Then after the recombinant DNA plasmid, means the gene of interest will be putting it into the recombinant bacteria, and then after the bacteria will reproduce the colon of a cells in a cloning vectors, and then after we get insulin at the end. So the applications of a genetic engineering in a medicine is first in insulin production, where in the past, as there was no genetic engineering, we are more not updated with the technologies and all those things. So at that time we use a cow or a pig pancreas to obtain insulin, to get insulin, but it has many problems. But from since 1982, as we get more aware about technology and all those things, we came to know that a human insulin gene were interested in bacterium E. coli to produce a synthetic human insulin, and which is used to treatment for an insulin dependent diabetic patient. Then after, it is also used in human growth hormones, where to treat a growth retardation, like a draftism peoples. It is also used in follicle stimulation hormone. It will also contain, it will producing form of follism injection, means it contains a follicle stimulating hormone, which is used to treat infertility in males and in females. Then other biopharmaceutical other biopharmaceuticals under the development through the genetic engineering includes nowadays is a anti-cancer drugs and possible vaccines for AIDS and malaria. So the genetic engineering plays here a major role in preparation of a vaccine, where the microorganism yeast is produced to virus antigen and use as a vaccine that stimulate the human immune system against the virus. And this procedure has been successfully developed, development of a vaccine against the hepatitis B vaccine, and which is nowadays widely used. Then the second example is genetically engineered vaccine holds a great promising for a future. So as nowadays we get to know about more awareness regarding the cancer, so Shri Priyam Narendra Modi ji has a in government PHC and CSA center. They gave a awareness camp regarding the same, that they developed one human papillovirus vaccine, which is used to prevent cancer, cervical cancer, as well as vagina cancers in a females more compared to male. It is also used in a males also, but the cancer type, it depends on the cancer type and cancer stage also. This vaccine can also use in males also, but nowadays they prefer more to aware about the cervical cancer and vaginal cancer, though. So they give a very free and a free cost to teens and adults through at the age of 26. So but nowadays, as the food will got an inorganic food we consume, so the menstruation cycle comes in a girl very early stage. So from 9 to 10 age years to girls to up to 26-year adult girl, they give this vaccine. Then after, genetic engineering vaccines may be used to prevent a disease that have a resistant to traditional vaccine, which include HIV, tuberculosis, etc. Gene therapy. So gene therapy is traumatic gene therapy. It is a desire to introduce a functional gene to body cell, which enable to perform a normal functions, thus providing correction for a genetic abnormalities. As we got to know about some disease such as vitiligo, which is from gene oriented, heredity oriented. So once we get to know that we have a vitiligo, so we can consult to doctor, and doctor, some of the doctors will suggest us for a gene therapy, where the gene of a vitiligo, means the body cells, myeloma cells were there, melanocyte cells were there to for color pigmentation, that will be very suppressed. So to separate. So at that time, white patches in the whole body will get. So that gene will be altered. Then after we get the normal skin very slowly. Then second is a targeting therapy. This will cause an individuals by targeting therapy used by the body cells such as bone marrow and blood cells. Gene therapy has been successfully used to treat a chronic lymphocytic leukemia. Means as I pasted the picture here, the normal blood look like this, and the leukemia blood, which is look like the whole blood will be of a white. In the very rare cases there will be present of a red color. So it is also used in that also. And in Parkinson's disease, Parkinson's disease means any kind of a involuntary movement which we not want to wish that our body do, but the body will without our notification, without our indication, any involuntary movement will be happening in our body is known as an Parkinson's disease. So it is also used in a genetic engineering. Genetic engineering used in a wide range, like in animals. Example, Aquarius advantages salmon, which is used to prepare for a faster growth and hormone. It is used in also agriculture field also, where there is a golden rice which will be prepared by adding the gene of beta carotene synthesis gene, which will be adding. And then after with the solution, with the use, but when we why they will be gene alteration will be done, because the person who is suffering from a vitamin D deficiency, they can eat golden rice and they can cure about with that too. Okay, then genetic engineering, so use in very wide range or a very vast range now, vast rate very nowadays. So this is all about it. Uh, Please subscribe to our channel Principal IICT for more pharmacy related video. If you want to get to know about more videos, do comment and watch topic on what topic you want to go. We will shortly make the video for that topic also. Thank you for listening.